Yes. So our discussion meeting class last Friday, I have it recorded, right? I'm going to send you guys the link. So we agree on a YouTube channel, right? So that you can go and watch. You guys can go and watch everything that we are doing. So let me use this. Let me use this advanced. Okay. Okay. Now, so I'm going to upload every videos. The one we had last week, Friday, is on that YouTube channel. You can go there and watch it. I will share the link to the channel with you guys. It's not compulsory to, it's just for you can just watch it and some other videos that may be educative on it anyway. So, how many of you design the drugs? Yeah, let me start. Let me ask. How many people designed his or her own drug? Or at least played with the with the what is it with popcorn. Please, if you do that, you can unmute. Don't signify, just unmute, because we have this on channel, so I don't want to be showing you guys face. Just for yeah, so just unmute. I want to know if how many of you did that.
I did try to think it a little bit.
Wait,
I did it.
Jennifer, did you do it?
Yeah.
Okay, Dave, you did. You designed your drug.
Yes, I tried.
Oh,
So
great.
yeah.
How about Jennifer?
Yes.
Okay. Anna, how about you?
Yes.
Okay. Jamie, did you do something?
Yeah.
Okay. How easy was it for you guys? Let me know.
It was a little weird at first to try to figure thing
Yeah,
out with Chimera
yeah.
or Gotcha.
Okay.
But
But don't forget, it still follows the same chemistry theory, right? Okay. So don't forget, let me just give you a recap of what we had last week. So we talk about AI, right? And we talk about the application of AI in different phase of life, right? And I showed you guys that under AI, we also use AI for biomedical research and, for instance, in drug discovery, right? So I made that clear to you. And I showed you that when you, as a scientist that want to design a drug against a particular target, it is expected of you to have understanding of the disease, right? And we said, okay, for instance, if you're looking at SARS-CoV-2, right, you must understand SARS-CoV-2. How does SARS-CoV-2 gain access to the body? How does it replicate itself to produce many copies and causes sickness, illness, right? And I showed you that there are different ways. It can be entry, you can target the entry mechanism, you can target the replication of the DNA of the virus, right? And stuff like that. So, and we said for this project, we are going to focus our work on SARS-CoV-2 protease enzyme. Protease enzyme. So, and I told you that when you are trying to design the drugs, there are different approach. You can decide to design the drugs for yourself, right? So I showed how to design a drug, how you can design a structure using PubChem last week, last week Friday. And I also showed you that you can go to database such as PubChem, right, to look for compounds. And I asked you guys to suggest a particular drug for us, right? So you guys suggested ibuprofen and aspirin. And I showed you how you can search for ibuprofen, aspirin, and other drugs, and how you can download these molecules, these small molecules. And I need to understand that when you are downloading drugs, you download the SDF and you save it in Mol2, the 3D format. And I also showed you, in case you could not download the 3D, maybe you only got 2D. You download the 2D and you can use the Avogadro software to convert the 2D to 3D. So how many of you downloaded installed the Avogadro and the Chimera software last week? How many people?
I did.
Oh, thank you,
Me
Dave.
too.
And Anna, how about you?
Me too.
Oh,
Yes.
Jerry, great. Oh, OS and Jamie, what about you?
Yes.
Anybody that, so I guess no difficulties with installing Avogadro and Chimera. So did you guys try to fiddle with the structures you design on Chimera?
Yeah, a little bit. A
Oh,
little.
great. Great. That's fantastic. That is fantastic. So today, I've shown you how you can get compounds, right? I showed you last week how you can get compounds. Now, one thing I want to show you guys today is this. If you remember last week, I said you can design a drug based on an existing drug. How many of you agree? So, for instance, so in this study, in this work, right? I remember I also mentioned that you can use what is called a reference drug. Reference drug, right? So reference drug will be the control and like an FDA approved medication already on the market. So one of the drug that is used for SARS-CoV-2, in treatment of SARS-CoV-2, right? For protease enzyme, for the protease enzyme is called lopinavir. So let's go to PubChem. So lopinavir is going to be our lopinavir is going to be lopinavir is going to be our reference drug, right? So the drug we want to test aspirin, ibuprofen, and the design drugs that we design ourselves, all those four, and see if those drugs can interact with SARS-CoV-2 protease enzyme and stop the enzyme from working. So if you stop the enzyme from working, then we can stop SARS-CoV-2, right, in the body. So we are going to use lopinavir as a reference drug, lopinavir. So let's go and, this is lopinavir. Let's go and download the structure of lopinavir. This is lopinavir. Lopinavir is an let me try to show you some information about lopinavir. Drug. It's an antiviral drug, right? Lopinavir is an antiviral drug. Let me try to give. This is summary of the drug. What is this? Lopinavir is an antiviral drug. It's an antiviral drug. So lopinavir is, if you look at this, you can read the information. Another thing about PubChem, it can give you description of whatever medication or drugs you are looking. So lopinavir is an antiviral drug, antiviral protease inhibitor. Yes. Don't forget, our target in SARS-CoV-2 is protease enzymes, the protease enzyme. So when you are looking for a reference drug, it has to be a protease drug that has been used in the past. So lopinavir is an antiretroviral drug, right? Inhibitor in the sense that it can bind to a protease enzyme and stop the enzyme from working. So if you're able to stop the enzyme from working, then you can stop the replication of that virus. So lopinavir is being used by HIV and other diseases. So it is already approved by the CDC. So that's why we are using it as our reference drug. And if you look at this, what are we looking for? We are looking for 3D structures. But unfortunately for lopinavir, we only got 2D, right? So I told you, so we have to download this 2D and we can open it on Chimera, on Avogadro, right? So let's go and open this on Avogadro. Let me go to my Avogadro. This is Avogadro, right? So let me open that lopinavir. Lopinavir download. Yes, generate it for me. That is the structure of lopinavir. So this is going to be our reference drug, like our control drug. So let's save it. Save as, don't forget, so let me give it a name. Let me go call it Lopinava. Lopinava is going to serve as our reference drug. Let me save it in the IARC folder. See, so I can close this now. So let me go and open it on Chimera. I want to believe you guys are following me. So Lopinava. I don't know why this, but I'm sure most of you guys, once you click on it, it opens directly. It's not asking you for this to choose. Okay. So can you see the structure of Lopinava? Right? Good.
Yeah.
Good. So we now have our reference drug. So when you are looking for drug to design, when you are looking for to design a drug, don't forget you need a reference drug. Then you can try your compounds. Your compound may be something you design yourself, like the one we did last week and the one you have done. Your compound might be maybe you just download another drugs online, right? Now, another idea I want to show to you guys is this. Now, I said something the other time that you can design drugs based on your knowledge of an existing drug, right? So we can there are some AI servers, right? That I can say AI, I want you to give me structures, compounds that are structurally related to Lopinava. So if Lopinava can treat SARS-CoV-2 and HIV protease enzyme, then I can generate structures that are similar to Lopinava, not exact Lopinava that are similar to Lopinava. One of the sites you can do that is we use what is called Swiss similarity. Swiss similarity, right? Can you see me? Yeah, that's it. Swiss similarity. So what that means is you can give this server. Oh, why is it showing that? Oh, what happened? Why is this? Reload. So let me check. Is there a network issue? So what I'm trying to do now, I want us to Swiss similarity. Oh, this is unbelievable. What happened? Oh, I think this website is not available now.
It worked for me.
It works for
Um,
you? So
yeah, the
probably
first one.
is my network here. Oh,
Yeah, that first one.
yeah. I'm surprised it's not working. It's not working. So Swiss. Okay, I'm coming. Let me go to this. Let me go to the parent page. Swiss. Why? Why is not showing for me? Oh, this should work. Just fixing there. I think this model 2 is not working. Just give me 1 second, guys. I think that means the issue is with my network. I'm trying to ping my network. Unbelievable. So why is it? Wow. What the button? Okay, let me try another browser. Let me try another browser. Just give me a second. Okay, let me see. Oh, still the same thing. Wow. So I'm trying to. Okay, no what? I'm going to show that. I'm going to show that later then. If that is what it's showing for. Okay, let's leave that. So let's go back to now. So this is, I think we have someone has a chat. Yeah, I tried this. It's not showing. Thank you for that, David. Why is it showing? Okay, it's not showing. It's not. So let's go back to our work now. Right. So let me charge this. So what I wanted to do there is I wanted to generate similar structures to Lupinava. But since that is not working, let's leave that. I'm going to show you that next time. So now, don't forget, we now have set of compounds we want to try against protease, right? So the next question you will ask yourself now is, how do we get our protein? So I ask you guys to read on protein structures. So the place where you can get your protein is what we call where to get your protein, right? Is what we call protein data bank. Protein data bank. Protein data bank. PDB. You might have heard of it before. So just type protein data bank. Protein data bank. Right? Then click on the first page. So this is protein data bank. This protein data bank is also, I think, is managed by NIH himself. So every proteins, every enzymes that you can see for any disease for human are on this data bank. So now, what are we looking for? What protein are we working on? Can anybody remind me? What protein are we designing the drugs against?
SARS-CoV-2.
SARS-CoV-2 what?
The protease enzyme.
Protease enzyme. Thank you. SARS-CoV-2 protease enzyme. So when you are looking for, you must know your target. So let's type. So you type SARS-CoV-2 protease. So don't forget to put SARS-CoV-2 because there is SARS-CoV-1, right? So SARS-CoV-2 protease enzyme. right? SARS-CoV-2 protease enzyme. SARS-CoV-2 protease enzyme. So once you have SARS-CoV-2 protease, so now look at this is protease, main protease SARS-CoV-2 viral protein. Let's click on that and let's see. Do you see that now? Now it gives us, can you see this? It says it has lot of structure. This is just page 1 out of page 25. So when you are searching for a particular protein on data bank, right? You need to, first of all, know the name of the enzyme you are looking for. This is SARS-CoV-2 protease enzyme. Other viruses also have protease. HIV has protease. SARS-CoV-1 has protease enzyme. MERS, Middle East Respiratory Syndrome, has protease enzymes. Even Ebola virus has protease. So almost all viruses has protease. So when you are downloading your protein from databank, you must specify the organism and the name of the protein that you are looking for. Let me give another example. If you are designing a drug, right, against maybe you are against human glucokinase, right? Maybe you are trying to treat diabetes and you are trying to look for a drug against glucokinase enzyme. If you are developing that drug for human, you have to put on PDB, I'm looking for human glucokinase, because apart from human, other organism, dogs, goats, they have glucokinase. So you must specify the organism and the name of the enzyme that you are looking for. Is that clear? So that you are not using the wrong protein. You are not using the wrong what? The wrong protein. So if you look at the first list here, we have room temperature institute SARS-CoV-2 main protein. Main, that M means main. That PIO means protease. Protease with bound. This is the name of a drug that is bound to this protein, right? So let's look at this one. So when you are doing this, another thing you need to understand is this. Look at this information. It's from, it was gotten October, it was in May 2022. The method is X-ray diffraction, right? Organism, severe acute respiratory syndrome, coronavirus 2, that's SARS-CoV-2. Macromolecule, 3C-like protease. Yeah, they call it that name also. So, so if you look at this is one. If you look at this also, this was uploaded 2022 also, right? Protease enzyme. So you can choose which one that you want. You choose which one that you want. So let's choose this one. Let's click on this one, right? So if you click on it, it gives you more information. There's no mutation, right? This is from E. coli was used to express it. So let me explain what I mean by this to you. For you to be able to get an enzyme, a virus protein, you have to produce it in an organism. So they use E. coli, a bacteria. So that's what you need. So let's go down and look for more information about it. So this is it. Enzyme, there is in A, there is 305 sequence. Can anybody explain to me what they mean by sequence? Now, what is the building block of every protein?
Amino acids.
Amino acid. So what this is saying to you is there are 305 amino acids that make up the protease enzyme that we are looking at. So different amino acids join through a peptide bond to give us a big protein, right? So it says mutation. That means there's no mutation on that. The original copy of SARS-CoV-2. There's no mutation on it, right? There's no mutation. Can you please give me 1 second, guys? So this is the number of amino acids that we have on that, right? So if you come down, this is just information about the protein, right? So apart from that, we also have some molecules on the protein. We have this is a drug that was being tested on that protein. These people too are testing this drug on it. Then these are just normal DMSO that was part of it. So let's go and download this structure. So now look at, I want you to think of this. So we want to use this structure. So let's go here, right? Click on download file, right? There are different formats here, but the one that you need is PDB format. PDB format. So you can click on it. Now let me now give you another way you can do that. You see, this is what is called this 7QP5. This is the PDB code for this thing, for this particular one. So if you don't want to download this thing, if you don't want to download it from Pochem directly, you can go to Chimera, right? So if you go to Chimera, you can go to File, right? Fetch by ID. Fetch by ID. Click on Fetch by ID. So make sure you click on PDB. Then you can paste the PDB code of that protein there. And don't forget, you must be connected to the internet before you can download directly from PDB. So once you put this, so don't forget this is the PDB code. Let me go back and show you. The PDB code 7QT5. That's what I pasted there. Can you see? 7QT5. So let me go back to this. So once you do that, click fetch. Because I'm connected to the internet, can you see? It gives me the structure of this, right? It gives me, this is the structure of SARS-CoV-2 protease enzyme. Now, another thing I want us to know is, this is usually a dimer. A dimer. So let me just stop like that. So this is the protein that we are looking for. So if you know the PDB code of the protein, you can fetch it directly from the internet. But if you don't know it, let me go back to that. If you don't know it, just come to this. You can just click download file, then choose PDB format. It will automatically, can you see? It will automatically download it for you. So let me copy it into our iAd folder, Control V. Right. So that is it. So now we've downloaded our drugs, right? The drugs I have designed, right? So the next thing is the protein. The next thing is what? The protein. And this is the protein now. The next thing is what? Is the protein. So you can also do this also at your own convenient time, right? This is the protein. So what do we want to do with the protein now? We want to do what is called molecular docking. What does that mean? Say molecular docking. Molecular docking is a technique, right, in computer-aided drug discovery or in AI drug discovery. So what molecular docking does is, from the word docking, when you dock something, that means two things come in contact and fitting in each other. So what we are trying to do to molecular docking is, we are trying to put every compound that we have downloaded, we want to be putting them at the active site of this protein. The one we downloaded, aspirin, ibuprofen, and the one I designed and you guys design, we will be putting each of them at the active site of that protein, right? So when we put it there, the computer will be the one to put it there and to generate for us a score. So that score is going to be a measure of how convenient, how fitting each of those drugs is at the active site of the protein. Let me come again to explain that again. In drug discovery, once you have your enzyme, you have your drugs, your compounds you want to test. The next thing is to do what is called molecular docking. So molecular docking is an AI technique, right? That is being done with AI softwares like Chimera. So what that does is we take every single drug and we put at the active site of the protein and see is the drug fitting well at the active site of a protein. Now, before I go further, you may be asking me, what do Dr. Dogu mean by active site? Active site, are you with me? Active site in an enzyme is the site where either a drug or substrate bind to for it to perform its physiological function. Let me give you an example. Last time I was telling you guys about key and lock. I don't know if you remember last Friday. Hello, are you still with me?
Yeah.
Please, I want your verbal response. I try to pick up the video so that you guys' picture won't be showing by the time we upload the recording. So just I want your verbal response. So I use an example of key and lock, right? Now, let me ask you this question. On your lock, there is a point on the lock where you must put your key. Am I correct?
Yes.
Now, but that point is not the only part of the lock. There are also some other part on the lock. If you put your key on other parts, apart from that hole that has been meant for the keys to be, will you be able to unlock the key?
No.
No. So for that key, for that padlock, for that lock, that point where you put the key is the active site of the lock. It is the site where your key can do the magic of opening the lock. So the same way happens to an enzyme. This is a SARS-CoV-2 protein. For any drug to be able to stop this enzyme, for you to be able to stop SARS-CoV-2, this enzyme, your drug must bind at the active site of this protein. So your drug cannot just bind anywhere. No. If it binds where it is not the active site, it's not going to have any effect. And that's why if you put your key in the wrong place on your padlock, it's not going to open until you put it at the right point on your padlock before you can open the lock. So the same way, for your drug to be able to bind and stop SARS-CoV-2, it must be able to bind on the active site of this protein. So just as you know the hole that is the active point of on your padlock, you must know the active site of this enzyme. So it is it is on the active site that you are going to dock all your drugs, the compounds that you have downloaded and designed. That is where you are going to dock them. And the Chimera software will now give you what is called a docking score. So that docking score will be the one to tell you if your drug is fitting well at the active site of your protein. Any questions so far? Any question from anybody? Jennifer? Anna? Do you guys have questions?
No.
Is this straightforward?
Yes.
Yes.
Okay. Okay. I'm glad to see that, to hear that. So every protein has an active site. So that active site is where your drug must bind to for you to be able to inhibit or activate the enzyme. So in this protease enzyme that we are using as an example, our objective is to inhibit this enzyme so that if we can stop this enzyme from working, then SARS-CoV-2 replication will what? Will stop and it won't take over the body. So the question now is, how do we know the active site of a protein? That is the question we must ask ourselves. How do you know the active site of a protein? There are different ways of knowing the active site of a protein. Oh, before we go there, let me quickly show you guys something. Forget this is a protein, right? And I told you proteins are made up of what? Amino acid. So all these things that you are seeing, they are amino acids, right? Oh, I think I should have given you an assignment. What are the 20 essential amino acids? Have you guys heard of 20 essential amino acids before?
No.
No. Oh, thank you. No, I think that is where before we go, maybe that's what I'm going to do before we go into that. So there are a lot of amino acids, right? But the essential amino acids that are major part of every protein, there are 20. There are how many? 20. So let me show you amino acid table. Yes. No, I don't want the coding. Amino acid. So there are 20 amino acids, right? Can you guys see my screen?
Yes.
Yes. There are 20 amino acids. Number one is glycine, right? Glycine. And in biochemistry, there are 3-letter code. There are 3-letter word for designating each amino acid. And there is also 1 letter. So for instance, glycine, the 3-letter code for glycine is GLY. GLY. The 1-letter code is G, letter G. We also have alanine. The 3-letter word is ALA, and the 1-letter word is A. So this is going to be an assignment for you. I want everybody to go, right, and study the 20 essential amino acids and their 3-letter words, 3-letter code rather. 3-letter code, yes. This is alanine. So we also have serine. Serine is SER, and the little letter is S. We have threonine, cysteine, valine, leucine, isoleucine, methionine, proline. I'm telling you, if you're looking at a career in medical school, biological science, there is no way you can escape this amino acid. If you are in fact, let me say, if you are going to science, biological science, you can't escape amino acid. So it's going to be a plus for you to, even if you don't know, if you may not know the structures, you can maybe just know the names for now. Valine, leucine. So these are the amino. So adenine, lysine, histidine, glutamine, asparagine, glutamic, aspartic, tryptophan, tyrosine, phenylalanine, proline, methionine, and the rest. Right. So those are the amino acids. Now, let's go back to our structure. If you go to, if let me put my cursor on this. If you look at where I put my cursor, what can you see? What 3-letter can you see?
VAL. VAL.
VAL. And that VAL means valine. That is the amino acid. Valine, amino acid. So it's valine on number 204. 204, right? So if you put it here again, this is another valine. Let me put another place. This is tryptophan. TYR, tryptophan. So anywhere you put, this protein is made of what? Amino acid. This is histidine. The 3-letter word for histidine is HIS. So that number is saying is histidine on number 246 of the O305. If you remember the sequence, Let me go back here again. This is another, this is tyrosine on 118. This is serine. Let me show you how you can. Let's go to residues. You see, can you see all of them here? Alanine, arginine, asparagine, aspartate, cysteine, glutamine, glutamate, glycine, histidine, isoleucine, leucine, lysine, methionine, alanine, choline, serine, threonine, tryptophan, tyrosine, valine. Look at it. So those amino acids are the ones that come together to make every proteins and every enzymes. Either the enzymes that metabolize the food that you eat, they are proteins. There is an enzyme in your saliva called an amylase enzyme, right? That enzyme is called amylase enzyme. It metabolizes when it is one that breaks down every food, every carbohydrate food that you eat. They are all made up of what? Of protein. So please, assignment for you guys, and you are submitting this particular one. You are submitting this particular one. Give me the first 20 amino acids, study them, and give me their 3-letter and 1-letter code. Take note of that. So I will be expecting that before our next meeting on Friday. So let's continue. So now, don't forget what you are trying to do. We are trying to do molecular docking. Now, we have a protein that we want to develop a drug that can stop it. And that protein is SARS-CoV-2 main protease enzyme. We have drugs that we want to test. We have aspirin, ibuprofen, and we have 2 drugs that we have designed ourselves. And we also have what is a reference drug, a drug that is already approved by FDA, that is being used to treat protease disease, protease enzyme of different viruses, and that is lopinavir. So that one is lopinavir is going to serve as our reference. So we have gotten our drug, we have gotten our protein. So, and I was trying to explain to you that molecular docking is you putting every compound that you have at the active site of the protein and be able to see which one is going to fit where at the active site of the protein. Because if it is not fitting well at the active site of the protein, it cannot stop the enzyme. Yes. And don't forget what we said last time. For your drug to be able to work, it must interact, if you remember, with the target. So the target is the protease now. It must fit complementary shape, right? And it must be able to exert physiological function. So now, docking will let us know if our drug is fitting, is interacting well at the active site of the protein. And I said to you, active site is the point on the protein where your drug can bind to, right? To either inhibit or induce the protein. So in our own point, we are looking for a drug that can bind in this protease work. We are looking for a drug that can bind at the active site and inhibit the protease and inhibit the protease enzyme, right? So that's great. Now, I said, how do we know the active site of a protein? That's where we're going to stop today. How do we know the active site of a protein? There are different ways to know the active site of a protein. Number one, you can read about the protein. What did I say? You can read about the protein. Let me give an example. You can ask, active site, you can read about active site, amino residues, amino acid residues of SARS-CoV-2 protease. Just put main protease enzyme. That's one way. So let's look for it. She's saying, which one is giving us? The main amino acid residues now this one is giving you the main amino acid residue in the active site of SARS-CoV-2 protease. So let's see what this publication is saying. right? So you can go down now. So what we want to do, these people also did it. So now they dock this at the active site, right? And if you look at this, let's go. I'm going to show you this, but I want to show you this, but I'm going to show you another simplest way to know it. Now, look at this. At a key 16 proteins, SARS-CoV-2 has 16 residues, but only 3 of them are exposed to solvent. Catalytic cysteine 145 is the most important cysteine located in the catalytic site. So when I'm talking about active site, I meant catalytic site. So what this is saying is, anywhere you see cysteine 145, that is where the active site of this protein is. So let's go and look for cysteine 145. So we can start looking for it. 145. We can start moving our cursor around, right? 145. Let's start looking for 145, right? 145 is 122. This 149, 148, 146. Okay, so it's probably, let's look for 144, 145. Can you see? And what is there? Cysteine 145. So the active site of this protein is what? Is here. So, but that may take at times that may take a longer time to get, right? You can use a command script, like a simple code, to know that. Go to general control, go to tools, general controls, command line. So when you click command line, it's going to give you this command script. And how do you do it? Is you want to select, you say select cell, a colon, comma. Don't forget what you are looking for is cysteine 145. Just put 145, right? And press enter. Can you see it highlight to us that this is what you are, this is, if you look, can you see this light? It selects it as cysteine 145. So what that is saying to us is the catalytic site of this protein is on this side. On this side. That is one approach. One approach that I've just did now is you can, so I want to take into that, you can do reading about the protein or type it in Google. What are the catalytic amino acids of your protein, whichever protein that you are working on, right? The whichever protein that you're working on, just put that and so you get it. Now, another approach to get the active site of a protein, which is one of the simplest way, to go back to protein data bank, where we get that protein data bank. What did I call it? So we go back to it. If you look at this one that we did, it tells us that this protease enzyme is bound with a drug. The drug is Z31 base, right? And if you come down, this is going to tell us the drug. Where is the drug? This is the drug, right? This is the drug. So the code for the drug is GWX, right? So let's go back to Chimera. Now, on Chimera, you can select a structure. So so with this one that so we know this is the active site of the protein now. Do you get it now? We all know this is the active site of the protein. This is the active site. So if you don't know, you can, since your drug has another drug, since your protein has another drug attached to it, you can come and you know the name, come to select, go to residues, and go to, now this is the drug that was attached to this protein. Let's click on it. This is the drug. So, which means the drug binds at the active site or the catalytic site. and look at it. If you look at what we did, the SL that we did, can you see the two of them is almost on the same place? So anywhere you find the drug bind to your protein, that is where the active site is. So this is the drug that bind to this protein, and this is the catalytic site residue. So this is the active site of the protein. So it is very good to know the active site of your protein so that you are not binding your drug in the wrong place. For instance, if we are binding our drug at this point, what you are getting is a false. It's going to be a false result because this is not the active site. So even if drug binds here, right? If drug binds here, let me say based on mechanism, if drug binds here, there is high probability, let me say like 80%, that drug is not going to work on this enzyme, right? But if it binds here very well, there is 80% that is going to work, is going to inhibit or induce this enzyme. So let me give a recap. Once you know your protein, the next thing is to identify the active site of your protein. And how do you know the active site of the protein? One, you can read about the protein. Go to Google, ask for the amino acid residues at the active site of the protein of your interest. In this case, I only type active amino acid residues at the active site of SARS-CoV-2 main proteins. If you are working with other enzymes like glucokinase, amylase, or you are working with other protein, maybe you are working with hemoglobin, right? Or you are working with cytochrome P450 enzyme, for instance. You just have to type amino acid at the active site of that protein. And by the time it will give you some manuscript that is related. And if you read through this manuscript, it will tell you the active site, like we did. So we did this one with search, and it made us know that cysteine 145 is anywhere you find cysteine 145, that is the active site of this protein. So that's one approach. Another approach is when you download your structure from PubChem, from databank, protein databank, you can, if it is bounded to a drug, an inhibitor, that inhibitor, where that inhibitor binds is likely to be the active site. If you are not sure, you can also read more about that active site. Are you getting the point now? So for this now, we have gotten the active site of the protein, right? Now, let me now say something. So what is next now is for us to do molecular docking. What do I call it? Molecular docking. That is what is next for us now. So to do molecular docking, we must first of all prepare our ligand on Chimera, prepare our protein also. But I'm not going to bug you guys with that today. That's where we're going to start next week. Or sorry, on Friday, right? So I want you guys to fiddle around, go to PubChem, download one or two structures, right? Protein, you can be on this one we just did now. You can also, maybe you can go and look for this structure, 7q-T5. Or you can look for other enzyme. If there is an enzyme you have been taught in school, right? Maybe amylase enzyme in the saliva, maybe glucokinase or any other enzymes that you have been taught in the past. Go to PubChem, type it there, right? But don't forget, specify the organism. Is it human? Is it virus? You have to specify so that you are getting the right one, right? So that's that for today. I want you guys to do that. And don't forget the assignment. I want you guys to go and read the 20 amino acids. Know them, right? It will really help you in understanding this process. If you have any questions, if you are sure to ask, you can put this in the chat.
I have a question.
Okay.
You said you wanted us to submit the assignment for the 20 amino acids.
Yeah, I want you guys to submit. I want you to submit that on or before Friday, 12 noon.
Okay, do we like email it to you?
Yeah, just email it to me. Yes, email it to me.
Okay. Okay.
Yeah. So let me.
Sir, what's your email?
Okay, let me put my email here. Yeah, that's my email. So the assignment is
Excuse me.
yeah, I'm listening.
So, like, all you want us to do is read it. Are we supposed to write anything?
What is it?
Are we supposed to write anything after we read it?
Yeah, just show me, just give me the information of what you have, just don't put the structure, just the name of the amino acid and their 3-letter and 1-letter code.
All
Right?
right.
So in that, I also now I want you to read about this. So that's the first question. The next question is, that's the next questions. So I want you to read about amino acid, right? And I want you to have understanding of how amino acid comes together to give protein a structure. So that's why if you read primary, secondary, and quaternary protein structures, if you understand what I mean, right? I want you to understand that is very, very key. In drug design, you must understand protein structure. You must understand protein structure, right? Any other question?
Oh yes, Mr. Kenny. So how would I format all my answers from the question? Like, do you want
I
me to
didn't
just
get you.
put?
Can you speak louder, please?
Yeah, can you hear me?
I
Can
can
you
hear
hear
you
me?
clearly now.
All right. Yeah, I said, how would I format my answers to the questions? Like, do I just put them all in, like, number them by one,
You just
one,
put
two?
number one and put your answer to it. Number two, put your answer below it. Yeah,
All right. Thank you.
just in Word documents. It's fine.
Okay, thank you very much.
All right. So on or before 12 noon on Friday. On Friday. So if you guys have time, go to Protein Data Bank, download structures yourself, right? And fiddle around. You will see everything is going to be on proteins. So next time when we meet, we are going to docking. Yes, we are going deeper into the work now. We are going to molecular docking, and that is where we are going to start seeing by the time we do it, I will ask all of you when we see the result, which of these drugs that we have designed and downloaded do you think is going to work against SARS-CoV-2? You are going to tell me. So that's what we'll start from there next week. Right? Any additional question? All right, you guys have sorry, let me quickly send you yeah, I will send this. Just give me 1 second. 1 second. Since we agreed that we should be putting our recording on what is it on YouTube for you guys to be able to look at it. Okay, so let me drop that for you guys. So please, this is just it's not the Profits YouTube channel. It's just for educational purpose and for if I should be sending you guys the videos of everything we make every day, it will occupy so much of space. So that's what my previous student suggested, and that's what we have been using. So if you go there, I'm going to upload. You will see our previous class on it. You also see to this class on it as well. You guys can go there. If you can watch anything, if you still don't understand, you can get back to me. I will put you guys through. All right. So it's just a YouTube channel. We may decide to subscribe or not. What I want you to do is you guys should just learn what we are teaching. If maybe you want to watch something again and again, just go there. You may subscribe, you may not. You are not any under obligation to do anything. We just watch that. All right. Okay. See you guys on Friday, right? So I'm also working
Yes, sir.
I'm also working on the payment of you guys stipend. So hopefully I should probably have something for you guys so that you can start the process. Okay. All right. Enjoy your days.
All right, thank you. You too.
Bye.
Bye.
Thank you.
You're welcome.
Bye.
Bye.
Bye.